Aloha and good morning. It's my great pleasure to have been asked. When they asked me to introduce Dave, I was like, oh my God, this is phenomenal. I've known Dave for about 30 years, and although I'm not sure Dave really needs an introduction anywhere, and especially here in Hawaii, where I think he is Mr. Ocean pretty much here. He's very well known. In fact, he just told me that on top of all the other things he does, he's involved in a partnership with the different businesses here in Hawaii to sort of bring scientists and tech to the general public. So that's another one of Dave's many things. Dave is a world-renowned microbial ecologist, a biogeochemist, and he's made major contributions in many different areas and worked in very many different parts of the ocean. He's been a long-standing member of ASLO and a great contributor and supporter of our aquatic and ocean sciences meetings whenever they're held here. And he's also a 1998 recipient of the G. Evelyn Hutchinson Award for his leadership in research, exemplary teaching, mentoring, and his general citizenship. And I also found out this morning that Dave is actually on sabbatical, and he's agreed to do this despite the fact that he's been on sabbatical. And he was, on top of giving this plenary lecture, he also was instrumental in getting the photo exhibit and the stories about the HMS Challenger's visit here to Hawaii. So I think we really owe Dave a debt of gratitude for all the things he's done for ASLO. Dave started in New York. He got his undergraduate degree at the State University of Buffalo in New York, and then, like many of us from those more northern climes, he moved south to Florida State to a warmer, saltier place, to Florida State University to get a master's, and then I think he saw the light and he started heading west. So then he went to Scripps Institute of Oceanography to do his PhD in ocean sciences. And then again, he keeps going west. It seems to me that he went right from there to come here to Hawaii, where he has been and rose through the ranks really rapidly. I think it was it really 5 years he went from assistant professor to full professor? I've known Dave a long time, but I really looked at his CV and it was like, really, 82 to 87, that was it? And so just look at this role model, you young people out there. It's pretty impressive. And Dave has served in many leadership positions in many societies, as well as an incredible list of, you really should go look at his CV on his website. He's had so many leadership positions here in Hawaii. And starting in 1988, this story that we're going to hear about Station ALOHA, he first started what was called the Hawaiian Ocean Time Series, which I think was funded for a few years and then eventually became a long-term ecological research project, which he later morphed into a science and technology center, which was Seymour, the Center for Microbial Oceanography Research and Education, and that was funded by NSF. And then after that, he's now gotten the funding he was a Simons Fellow to keep things running, because the NSF STCs only have a lifetime of about 10 years, and then they expect you to transition. They want you to keep that Science and Technology Center going, and Dave's one of the, I think, again, another one of the rock stars. He's actually had funding from the Moore Foundation, and now he actually has funding from the Simons Foundation to continue that work at ALOHA. So he's, again, and I was involved in an STC, the Center for Deep Energy and Biosphere, and Dave was one of those people when we were up for our renewal. I got in touch with him and he just shared so much information about what you do to make sure you get renewed. He was just incredibly helpful and gracious. So again, thank you, Dave, for everything. And Dave served on over 70 major oceanographic expeditions. He's actually made 30 dives in Alvin. He has over 400 publications and over 70 research grants. He's worked around the world. He's done a lot of research in Antarctica early on. He's worked all over the Southern Ocean. He's worked in the Black Sea. There's so many places. It's unbelievable. And again, so many different awards. I think he has every named award in ocean sciences. As I said, he got the Hutchinson. He also got the Bigelow Medal from Hui. He also had gotten the Huntsman from the Royal Society of Canada. He got the Cozzarelli Prize and the Agassiz Prize from the National Academy. And most recently in 2015, he was awarded the DuPont Industrial Bioscience Award from American Society of Microbiology and the Balzan Prize in Oceanography. He is a member of the National Academy of Sciences and also a member of the American Academy of Arts and Sciences, which includes people from all different walks, like I think Bruce Springsteen's a member of the AAS, I think. I don't know who was there when you became a member. So Dave's been amazing, and I'm actually not sure where he ever finds time to sleep or ride his beloved motorcycles. So with that, I'll...
Well, thank you, President Duguay, for that very generous introduction, much of which you said was correct. And I should add that Bob Dylan and Paul McCartney are also members of the American Academy of Arts and Sciences, and they were two of my childhood heroes, of course, growing up in the 80s. Well, I'm delighted to be here today to speak to all of you about Station ALOHA, a gathering place for discovery, education, and scientific collaboration. And I'm really just a spokesperson. This is a major collaborative effort that involves many scientists around the world. And I'm sorry about the rain, but as we heard on the plenary on Sunday, rain is good. It's all about life. And this is what keeps the mountain to the sea, the watershed, the Apua'a, keeps it going very strong here in Hawaii. So the rain is good. Please get out and enjoy it. It's not cold rain. I heard there's a blizzard warning on the Big Island for the slopes of Mauna Kea and Mauna Loa. So if you miss the snow from back home, you can grab your shovels, and it's only a short air trip away, and you can go enjoy yourself on the slopes of Mauna Kea. Okay, I'd like to make a few acknowledgments up front because it's very important that I do so. And I'd first like to acknowledge the ASLO leadership and the organizers of this meeting. Events like this don't just happen. They require careful and comprehensive planning and skillful execution, both of which are very evident in the meeting. So I'd like to thank Kathleen and Paul and all the leadership of ASLO for that. I'd like to thank all the past and present collaborators and people who have worked at Station ALOHA. Whether you've been there or not, you maybe have worked with some of the data sets and published some interesting papers, so I'd like to acknowledge that. And especially all of you for making a port call in Honolulu. And then the funding agencies that have sponsored our work, the Simons Foundation, the Agaron Institute, the Gordon and Betty Moore Foundation, the Balzan Foundation, and of course NSF, who has been funding Station ALOHA since its inception in 1988. And I would like to thank all of you and your sponsors. We heard on Sunday from President Duguay that there's 50 different countries represented here. And all of you are here probably because of funding from your local or national science foundations. So thank you for funding and for support of this meeting is truly an international thank you. Speaking of port calls, I just wanted to bring your attention once again to this art exhibit that Drew Harvell and Peter Williams put together, the bizarre and the beautiful plankton, these beautiful images, lithographs from Ernst Haeckel's work. And we also have a Challenger Honolulu port call exhibit there, which shows, among other images, the image of King Kalakaua, David Kalakaua, who was the king of the Kingdom of Hawaii at the time, who actually came and paid a visit to the vessel. And this great auditorium is actually named in his honor, the Kalakaua Ballroom. Mark Twain, also known as Samuel Langhorne Clemens, visited the islands and didn't quite have this view that we see from the International Space Station, but he traveled around the islands, and this is a quote from one of his newspaper accounts back to the Sacramento Bee: "The loveliest fleet of islands that lies anchored in any ocean." And I think you will all agree, especially looking at an image like this. And you know the theme for this meeting is Mountains to the Sea. I like this little tag, M2C, Mountains to the Sea. I'm going to talk about a slightly different aspect of the M2C. We have a word in Hawaiian called Makai, which means to the sea. So this talk is really the other M2C, Makai to the sea. And I would like to present some of the work that we've been doing out in the open ocean at Station ALOHA. This work is a collaborative work and a work based on complementary programs, including the Hawaiian Ocean Time Series program that Linda mentioned, established in 1988, shown here at the top. The Center for Microbial Oceanography, Science and Technology Center, funded by the National Science Foundation, which was established in 2006. And more recently, the SCOPE program, the Simons Collaboration on Ocean Processes and Ecology, that was started in 2014. If you look at Station ALOHA, we have a number of different types of research that goes on there. And it's shown here, this red circle at 2245, 158. And just below that, there's a little yellow box that says ALOHA. That stands for a long-term oligotrophic habitat assessment. And I consider that all of the ship-based work, the satellite-based work, and the remote sensing work that's being done now by in-ocean drones. There is also the WATTS program, the Woods Hole Hawaii Ocean Time Series study, which maintains a meteorological and physical oceanography mooring at the site, funded by a National Science Foundation and NOAA, run by Bob Weller, Al Pluteman, and Roger Lucas here at the University of Hawaii. Then there's the ACO, the ALOHA Cabled Observatory, shown here in cartoon with this yellow line that goes from Makaha on the island of Oahu all the way out to Station ALOHA. In fact, that's a fiber optic cable, a high bandwidth, high power cable that allows us to maintain a bottom observatory, and that's currently run by Bruce Howe, who is here today. And then we have the deep sediment traps, which is an independent enterprise run by the Gordon and Betty Moore Foundation. Now, in 2015, we were honored at Station ALOHA by the American Society of Microbiology, the largest life science organization in the world, some 60,000 members, as a milestone in microbiology site. This is one of 15 sites around the world that was so designated because of its historic and visionary contributions to the field of microbiology, in our case, microbial oceanography. And we know that we have the mountains. Mauna Kea, as Linda told us, was the highest mountain on Earth, some 10,000 meters. And we have the deepest observatory, the ALOHA Cabled Observatory. And this led Victor Hugo, this is a bit anachronistic, but it led Victor Hugo to say, where the telescope ends, the microscope begins. And he gave this rhetorical question from his epic work, Les Miserables: which of the two has the grander view? And as you see on the left-hand side is the summit of Mauna Kea, where we have multiple observatories looking out into outer space. And what's shown in the image above that is an image of 12 billion light-year corridor of space, showing all the stars and planets and galaxies. And shown on the right is the inter-ocean observatory, taking a look through seawater, 12 billion microbes per liter. And those two images could probably be interchanged, and only a couple people in this room would be able to correct me on that. It's an amazing symmetry, looking out into outer space and looking down into inner space, that the images are essentially very similar. Now, this is what I'd like to cover today, today's discussion points: what matters and why. I'd like to talk a little bit about microbial oceanography and try to convince you that it is truly a sea of opportunity. Then I'd like to talk about our planet that we all share, and especially the human imprint on our planet. And then talk finally about the role of time series as sentinels of the sea. First of all, I'd like to talk a little bit about sea microbes and their diversity. And as Louis Pasteur said many years ago, the very great is achieved by the very small. This is a slide on the key concepts of microbial oceanography, taken from a brochure that we prepared in the Center for Microbial Oceanography of the same name, Key Concepts in Microbial Oceanography. And this is available on our website. It's got a nice centerfold. You can download it and hang it up in your children's bedroom or in your grandma's parlor, and they can learn all about the key concepts of microbial oceanography. Here they are in a nutshell. Microbes are small, abundant, and ubiquitous. They're ancient and have an enormous genomic potential. And truly, that's what keeps our planet habitable, as you'll see in a few minutes. And many recent discoveries have led to new paradigms about microbial life in the sea. For example, I call this collective new set of information and discoveries the second golden age of microbiology, following on from the first golden age of the mid-19th century, where we found what microbes really were and what they did: disease and infection, fermentation biology, debunking spontaneous generation. This truly is equivalent to that in terms of its impact on the way we think about our planet. It is the second golden age of microbiology, from 1988 to the present. So these discoveries have all been made since we established Station ALOHA in 1988. I'll just give you a couple examples. In 1988, the same year that the HOT program started Station ALOHA, Penny Chisholm, Rob Olson, and their colleagues discovered Prochlorococcus, which we now recognize as the most abundant plant on our planet. I would submit to this august group of scientists that if we didn't know about the most abundant plant on this planet, then we didn't know too much about the way our planet operates. This was discovered only 30 years ago. And Lisa Campbell, 2 years later, published some very interesting work from Station ALOHA, confirming Chisholm's observations from the Atlantic, showing that at Station ALOHA, Prochlorococcus was also indeed the most abundant organism, thereby showing that it was really a transoceanic and cosmopolitan phenomenon. Then there's a bunch of other first-order discoveries that I don't have time to really talk about, other than to say that some of these discoveries by Ed DeLong, who's been a long-term colleague at Station ALOHA, the discovery of planktonic archaea, for example, the discovery of proteorhodopsin phototrophy, these have fundamentally changed the way we think about the flow of carbon and energy on our planet. And then, of course, since 2005, since the first metagenomic discovery in the ocean, we now have the omics revolution, or evolution, because it seems to keep changing. New genes, new organisms, and certainly new research opportunities for all of our up-and-coming graduate students. And it's always amazing to me that nature could essentially write equivalent of an unabridged dictionary of life based on just 4 letters: A, T, G, and C. It's truly amazing. Now, not only is there genomic diversity in the microbial world, but there is metabolic diversity, which scales from the genomic diversity, of course. This is really the phenotypic expression of all the genomic potential on the planet. And we know that all life in the ocean or on land requires a source of energy, requires a source of electrons, and requires a source of carbon to make a living. Conveniently in the ocean, we have a multiple array, a smorgasbord, if you will, of supply of carbon, energy, and electrons. And I show that in this metabolic menu on the right-hand side. And some of these caricatures are wonderful, magnificent marine microorganisms. And you can see that maybe one microorganism is a phototroph, one is a chemotroph, but to a large extent, most of the microbial life in the ocean has a mixed metabolism. We call that mixotrophy. It takes its meal from a number of different sources that are available to it. And this really creates a very, very intricate but complex web of life in the ocean that we're trying to understand. Now, diversity has, as I mentioned, phylogenetic basis. There is a metabolic basis. There is also a habitat or niche space diversity that we can speak about in the ocean. And the critical thing in all of these different types of diversity is time. So I'd like to tell you a little bit about time and take you back in time. I'd like to go back to 4.6 billion years of Earth's history and compress it into a single 24-hour clock, a nautical-style clock that you might see on a ship. And I'd like to look at a few of the important benchmarks in this history of our planet. Well, if the planet began at 000 on this clock, it took about 2 hours, 2 hours and 50 minutes to be exact, for the continents and oceans to form. So it took that long for the hot dust cloud to condense into our known planet today with its crust and lithosphere and its oceans and atmosphere. Shortly thereafter, we have evidence of the first bacterial fossils. We don't quite know the steps between 2:55 and 5 in the morning, but we do know that microbes, there were prebiotic synthesis, and microorganisms evolved perhaps in a warm, shallow ocean, perhaps somewhere else. We don't have much information about that, but we do have a fossil record dating back to about 3.8 billion years, 5 o'clock on this 24-hour clock of life. Those microbial life forms were all anaerobic. There was no free oxygen on the planet. And it took another several billion years for free oxygen to be formed. And that formed through a series of genetic mutations and selection for oxygenic phototrophy, which split water and made free oxygen during the process that we now recognize as oxygenic photosynthesis. This happened about 8:30 in the morning, right when the sun was coming up conveniently for photosynthesis. Then oxygen started accumulating on our planet, and by noon the oceans were rusting. The reduced iron supply was turning into ferric oxide and depositing in the seabed and on uplifted terrestrial environments, leaving a geologic record of that time so that the geologists can go back and date those red beds very accurately, so we have a fairly good timestamp on that. And then let's move ahead to nearly 2100 hours, 9 o'clock at night, the first macroscopic life. So the entire planet, the history of our planet, from 00 hours on a 24-hour clock to 21 hours, 75 or 80% of Earth's history was microbial. And it had a lot of time for microorganisms to evolve, to diversify, and to move into all sorts of very interesting niche spaces that we're just now learning about. I should say that humans, all of us, Homo sapiens, our great species, came on board about 50,000 years ago at 23 hours, 59 minutes, and 59 seconds. We've been here 1 second, ladies and gentlemen, on this 24-hour clock. And look at what we've done in 1 second. All the good things and all the evil things. Now, William Shakespeare wrote in his epic work, Julius Caesar, "The evil that men does lives after them, the good is oft interred with their bones, so let it be with Caesar." You all know the stanza. So let it be with humankind. This is an example of the good. I'd like to start with the good, start on a positive note. These are all the iconic contributions that we have made in science, engineering, and the arts. And many of these are well known and well recognized to all of you. It's the evil I'd like to talk about now. The evil that men do lives after them. And this is a paper, a very important classic paper, published in Tellus in 1957 by Roger Revelle, the giant of oceanography, and Hans Suess, on the increasing burden of carbon dioxide in the Earth's atmosphere. And this led to the International Geophysical Year, 1957, where Revelle was in charge of the oceanic contribution to the International Geophysical Year, the IGY. And they made a series of little booklets. This one is Planet Earth. They have one called The Oceans, one called The Land. I read these as a young kid in school. I still have my copies. In fact, this image was taken. I scanned this from the copy that I have. The mystery with 100,000 clues. This is Earth. This was to try to understand the Earth system as we now know it. But an interesting quote from this paper: "Thus, human beings are now carrying out a large-scale geophysical experiment of a kind that could not have happened in the past nor be reproduced in the future. And that led Roger Revelle and Hans Suess and others at Scripps Institution of Oceanography to do something about it. Let's start monitoring the Earth. And they turned to Caltech and to a very clever scientist named Dave Keeling, Charles David Keeling, who they hired to set up a series of observatories, one here at Mauna Loa Observatory, the first one, and observatories around the world to start looking at this increasing burden of CO2. And this is a well-recognized curve to all of you, or at least it should be, the so-called Keeling curve, which is the definitive evidence that humankind is doing something to the planet that we could never have done in the past and hopefully never be reproduced in the future, beginning the Industrial Revolution and piling up CO2 in the atmosphere. And shown in the upper left-hand side is a graph of the population of our planet from my birth year, 1950, to the present. It has increased nearly threefold, and it's not hard to make the connection between people on the planet and CO2 in the atmosphere and the total domestic or the total gross production on our planet shown in the lower right-hand side. So this is really a manifestation of the great standard of living that we enjoy on this planet Earth with the large number of people we have. And for those of you interested in learning more about Keeling, there's a wonderful autobiography that he wrote, somewhat in a bitter tone, called Reward and Penalties for Monitoring the Earth, that was published in the Annual Review of Energy and the Environment. And these are a few of Keeling's accolades. He was a National Academy member. He won the National Medal of Science from the President and won the Tyler Prize, this prestigious prize given by the Tyler Foundation at USC. But in his paper, he talks about some of the penalties of getting involved in running a time series program. He was criticized early on because his so-called routine measurements of carbon dioxide in the atmosphere strayed from basic science. But ladies and gentlemen, science, society, and sustainability are inextricably linked. And in fact, this is really the larger context. It's really what matters. This is an article taken from The Economist, Welcome to the Anthropocene. This is the age, the geological history of the Earth that has been influenced by humankind. Humans have changed the world. Now we must change the way we think about it. About this time, we started the time series program, and shortly thereafter, John Magnuson, an ecologist at Wisconsin who used to work here at the University of Hawaii, a fisheries biologist, wrote this wonderful paper in Bioscience called In the Absence of Time Series. The paper was called The Invisible Present. And his point was that without time series information, you are locked in an invisible present because you cannot put contemporary observations into a contextual past or predict their future. So he says here, this is a quote from his paper, in the absence of time series data, contemporary field observations are hidden in the invisible present. And this was indeed an assurance that we were on the right track when we started the time series program, and it was a call for arms for additional time series. So what do you measure, and where do you begin a time series program? This was a very difficult thing to decide. Leonel Weedy yesterday mentioned the JGOFS program, which was started in 1984 by the National Science Foundation. Among other things, this new wave of ocean science created some time series sites, two of them in oligotrophic gyres, one in the Atlantic near Bermuda, one in the Pacific near Hawaii, the so-called Hawaii Ocean Time Series program, and the Bermuda Atlantic Time Series Study, or BATS. These were the oligotrophic ocean twins. And oligotrophic waters are those that are very blue, shown on this satellite image from space of sea surface chlorophyll. Very low chlorophyll, very low nutrient concentration, very low productivity. But as you can see here, they cover a large percent of the Earth's surface, so they're very important for Earth system science. The Hawaii Ocean Time Series program, which started in October of 1988, has been going to sea on approximately monthly intervals, collecting core measurements that are publicly available at this website on the bottom. We have our data served in near real time with a lag of 6 months to a year for most of the data sets. Some are presented in real time. And we have a special focus on carbon, nitrogen, phosphorus cycling, and of course on microbial community dynamics. Lionel mentioned that he was in diapers while I was in high school. Here I am in the middle here, Dave. I wasn't really in high school, but this was the original group, Roger Lucas to my right, Eric Fearing to his right, and Chris Wynn and Steve Chiswell, who did a lion's share of getting the program started from the ground up. This is a photo taken just before we deployed on Hot 1, where Roger Lucas and I served as co-chief scientists, and I should say that this month we're going to be sailing on Hot 291. Makai to the sea, that's the M2C. Jagoffs is the joy of going out from shore, which is all what we love to do as oceanographers. And I have this quote on the bottom: when men and women like sea life, they are not fit to live on land. Samuel Johnson. Well, what were the major achievements of the Station ALOHA? I really can't summarize those in a talk like this. We have over 750 peer-reviewed publications. We have those listed on our website, and as I mentioned, we have a publicly available data set. But I should say that we have a high-quality 28-year climatology of many core parameters in the ocean to see how the ocean is changing or not. We have a soon-to-be-released ALOHA picoplankton gene catalog with 9 million unique protein-coding genes, along with their taxonomic origins, functional annotations, and variation over time and space. That's Ed DeLong's group. And we have, of course, new views on life in the sea. And every day here at ASLO is ALOHA Day. We've got a special session later today with oral and poster sessions. And every day this week and throughout the week, throughout the rest of the meeting, there will be some reports from data collected at Station ALOHA. And as I mentioned, we have our data freely available, shown on the bottom left. There's the website, Hahana. The data are also mirrored at the BICO-DEMO, the Biological and Chemical Oceanography Data Management Office, funded by the National Science Foundation and housed at Woods Hole Oceanographic Institution. And as of December of last year, there's a newly launched product which brings in 5 major databases all into one, into what they call thematic collections. And this is a screenshot of their so-called Hawaiian collection data set, and you can see our cruise track going from Honolulu Harbor out to Station ALOHA, and that was launched just recently with support from NSF, Scripps Institution of Oceanography, and EarthCube. So I would like to focus on a couple selected studies. I don't have time to talk about many. First is energy capture and export, net community production, and the aerobic production of methane. Here is a paper soon to be released. I think it's up on the online LNO website, the greatest journal on the planet, of course, ahead of print. This is by Ricardo Letelier and colleagues, and he has constrained solar energy input to Station ALOHA by looking at pigment absorption and the light field at Station ALOHA. And we estimate, based on his analysis, that somewhere around 8,000 calories per meter squared per day of energy enter the sea surface. And recently we've been looking at energy flux leaving the euphotic zone by collecting particles in sediment traps and measuring their total caloric content. This is a measurement that's never been done in the ocean that we know of. And what's shown on the left-hand side is a graph of the flux attrition of caloric content of particulate matter as a function of depth, with primary production bringing in about 8,000 calories per meter squared per day, and about 6% of that leaving the euphotic zone to feed the entire rest of the water column and the benthos behind it. And on the right-hand side, we have the carbon-specific caloric content, showing this very high caloric content in the plankton, some 10.5 calories per milligram of carbon, and as a function of depth, that energy is just taken out of the particles by the microbial decomposition process. And by the time you get down to 500 meters, there's only about 20% of the specific caloric content of the particles remaining. And we have no idea what those low caloric content particulate matter compounds really are. And this is a work underway. Another thing I'd like to mention is the metabolic state of the ocean. For those of you who are aware of this, there's an ongoing debate in the oceanographic literature. There was an entire 2 articles in the Annual Review of Marine Science, one by Williams et al. Stating that the oligotrophic ocean is autotrophic, the dichotomy by Carlos Duarte et al., former president of this august organization, claiming the oligotrophic ocean is heterotrophic. They both had asterisks, and the asterisk that I would like to say, or is it? And you might want to go to Sarah Ferrone's talk today at 10:45 to learn more about this, because I don't have a lot of time. But basically, if one looks globally at the rate of primary production shown on the y-axis versus the rate of microbial respiration shown on, production on the x-axis and respiration on the y-axis, with the dashed line being the 1-to-1, so that would be a unique metabolic balance where production and respiration equal each other. You can see that all the low productivity regions, these so-called oligotrophic ocean regions of the world, are above that line in respiration. So there's a greater amount of respiration than photosynthesis, leading to the conclusion that the oligotrophic oceans are net heterotrophic, which cannot be based on other information we have. So there's something wrong here. Either our conceptual view of the way energy and carbon flow through the system, or perhaps this is a methodological issue. Again, The Economist wrote an article about 20,000 colleagues under the sea, how you can bring robotics to the ocean and have drones collecting data on your behalf. We have done this, and we've brought a fleet of Seagliders out to Station ALOHA, looking at what we call the marine environmental sequencing. And ironically, it's the same 4 letters as we had in this wonderful dictionary of life: A, T, G, and C. These Seagliders can get absorption of light, looking at colored dissolved organic matter. They can get temperature. They can get gas like oxygen, which is what we're interested in. And they can, of course, get chlorophyll. So this is kind of a double entendre on that. Well, Dave Nicholson wrote a wonderful paper in 2015 where he put one of these Seagliders out at Station ALOHA, and lo and behold, he saw a wonderful diel cycle in oxygen, and was able to use that diel cycle, and along with a model constraining sea air flux of oxygen and diffusion exchange with the lower water column and the light level, and he fit this to a very nice sinusoidal curve, assuming a constant respiration. He was able to derive gross primary production, respiration, and net community production remotely with an in-ocean drone. Now, this is pretty remarkable, and we thought this was a phenomenal breakthrough until we went to the ASLO 60 for 60, the brand new issue that came out celebrating 60 years of the journal, L&O, and having the top 60 papers. And one of them, if you go back to 1956, this is exactly what Howard Odum did in flowing waters. Of course, he wasn't working in these very low productivity waters. He was working in Silver Springs and other places, coastal regions. But we essentially reinvented what Howard Odum had already done. And now we have long 3-month duration expeditions of these Seagliders, like this one shown here, Benedetto Barone, and he'll be giving a talk at 10 o'clock today in a session on some related work. But what he has done is to show in the black, this is the positive production of oxygen, so this would be gross primary production in oxygen units, and the red is respiration, and he's paired these on each day. And you can see that there's an enormous day-to-day variability in this so-called constant oligotrophic ocean of something as fundamental as gross primary production and respiration. We're trying to understand what causes a nearly 2-fold, on a day-to-day basis, change in primary production. And finally, this study of the aerobic production in the sea. I was so glad to see so many papers at this meeting talking about phosphonate cycling and phosphonate degradation. This is what I call from a paradox to a paradigm. Dan Rapetta and his colleagues published this paper late last year. And this is a true collaboration that could never have been done by any one group. And it's a collaboration between biogeochemistry, organic chemistry, and microbiology. And I point you to a few additional papers on this subject. So what are the time series challenges that we're left with today? The one interesting thing is the paradigms seem to keep changing. We keep learning more, and that creates a real problem for looking at long time series. Changing methodologies, of course, is something you need to be aware of. You don't want to change methodologies in the middle of a time series, but you don't want to avoid using improved methodologies. And, of course, changing mean states. And I point to the IPCC, the Intergovernmental Panel on Climate Change, AR5. This is the Assessment Report 5, published in 2014. It turns out, coincidentally, the Assessment Report 1 was published the year that Station ALOHA got started, 1988, and they periodically publish these reports of our planet. And not until this report came out did they even have a chapter on the oceans, if you can believe that. So I was privileged to be a co-coordinating lead author with Hans Portner on this chapter, and our executive summary, you can read it there. Ocean ecosystems have responded and will continue to respond to climate changes of different rates, magnitudes, and durations. Virtually certain. One of the iconic images of the ocean, and this is not a model output, this is not fake data, this is the real observation from Station ALOHA, resolved on a monthly time scale from 1988 to the present. Shown here in the red is the Keeling curve on Mauna Loa, the increase of CO2 in the atmosphere. Below that is the ALOHA curve, the PCO2 in the underlying water, showing the same rate of increase in surface ocean PCO2 as we see in the atmosphere, and then the corresponding acidification of the ocean. This work was published in 2009 by John Doerr and colleagues in the Proceedings of the National Academy of Sciences, and the following year won the Cozzarelli Prize for the best paper in physical sciences in the previous year. So we're very proud of this work. Now, one of the problems with the ocean is that there's a lot of things changing. It's not just acidification. The temperature is changing, stratification is changing, nutrient loading. And Philip Boyd, who will be giving the Hutchinson Medal talk tomorrow, I guess now, Paul, since he's stuck in an airport, published this wonderful paper about how we really need to simulate the change and future states of the ocean, looking at more than just single stressors. You have to look at multiple stressors, and this was the point of his paper with his colleagues, published in Nature Change, Climate Change. And the American Academy of Microbiology, the honorific wing of the American Society of Microbiology, is also interested in this, and they recently put out this report incorporating microbial processes into climate models. It was co-chaired by Ed DeLong, who's here in the audience, and Carrie Harwood from the University of Washington. This is really a report about why microbes matter, and specifically why marine microbes matter. And more importantly, that there's this large gap, this almost daunting gap in knowledge between climate modeling and biogeochemistry of the ocean. And we really need to work on that with great alacrity if we hope to do something about it in the future. So we believe that our collective work at Station ALOHA, shown by this little cartoon of the ship and the microscope and the islands, is sitting there clearly in Pasteur's quadrant. This is a wonderful book written by Stokes on the difference of really the continuum between basic research that is a quest for new knowledge, like Niels Bohr here in the upper left with his work on high-energy physics, and then the use-based research of Thomas Edison in the lower right, some 2,000 inventions in his lifetime and important patents, and then, of course, Pasteur sitting in there in this sweet spot between the basic and applied. And we believe that our research at Station ALOHA is bringing basic research. It's creating and disseminating knowledge that can be used, hopefully, to improve models, to inform society, and ultimately to protect the sea. So what is at stake? I believe the conceptual understanding of the sea is at stake, global productivity and other ecosystem services, ocean carbon sequestration, indeed planetary habitability and perhaps ultimately our own human survival. So quite a bit is at stake, and I think we need to move on this, again, with great speed and urgency. So finally, 4 lessons that I've learned from our Station ALOHA 1988 to the present is that you should focus on basic science at the cutting edge of the discipline. You should engage the best and the brightest collaborators, people who are interested in working together to make something larger than the sum of the parts, who want to share ideas and expertise and eventually data. And remember that basic science is partly discovery-based, and as Louis Pasteur proclaimed, chance favors the prepared mind. We should expect the unexpected. And we need to take risks and not be afraid to fail. But you better succeed more often than you fail. That's what David Packard once said. So finally, I'd like to leave you with this wonderful quote from the giant, Van Niel, who was a premier general microbiologist. He brought the field of general microbiology from the Delft School in Holland to this country, the USA, in the 1930s. In essence, science is a perpetual search for an intelligent and integrated comprehension of the world we live in. Thank you very much. Okay, thank you. Sorry I went over.
We have time for a few questions.
You want to come to the mic? Thank you.
Andrew Ross from Fisheries and Oceans Canada. I just wanted to thank you first of all for a wonderfully entertaining and thought-provoking presentation. You're mentioning John Magnuson's call to arms about time series and all of the science that you've shown. Suggested to me I should probably take this opportunity to give a shout out to the International Group for Marine Ecological Time Series, which is an IOC working group set up to write a report that synthesizes all available time series data from across the world. And of course, HOTS is a very important contributor to this because of the duration of that program that's been going on for ALOHA for 28 years. And another is our own Canadian Line P program, which has actually just celebrated 60 years. So if there are any policymakers in the audience, I just want to support our speakers' comments about the importance of time series, and we need to maintain these to keep pace and understand the amazing, fast and profound changes that are happening on our planet at the moment.
Well, thank you so much for that thoughtful comment, and it is a call to arms, so thank you, and we hope that other time series will get started this decade.
Yeah, Dave, that was stunning, as everyone would expect, and I just am really impressed with the fact that something that has such a high amount of inertia as the North Pacific Ocean can be used to show over long and short time scales these significant changes in physiology, metabolism, and geochemistry. And one of the things that I'd like to encourage among the audience is time series on smaller systems that respond differently, because the comparison with large systems, when something happens at ALOHA, it's big. And when something happens in a small lake, it may not be big, but it may be an early harbinger of something that is. And so I'd like to encourage, even if you started time series now in your professional lifetime, you could get one that's a lot of fun. And so I'd like to encourage young people to follow even 1/10 of your effort and great accomplishment to start a lot of time series.
Well, thank you for that kind comment, Russell. It's right on, and if I ever run for president, I'll have you as my campaign manager. But you are absolutely correct that scale does matter. All scales matter. And I think small lake time series are critically important and could be even more important than large ocean time series for just the reasons you said. They're harbingers for the future. And the more eyes we can have on this planet, the better off we are as humankind. Thank you very much for that comment. And I think we're going to turn it back to Paul to call for the break to get people going.
But let's thank David once again for a great talk.
Thank you. Sorry, I wanted to have a little bit more time for questions.